Hello and welcome to another video. In this video I'm going to talk about the epithelial tissue. So as you probably know, the body consists of four types of tissues. We have epithelial tissue, nervous and muscle and connective tissue, and each of these tissue types have undergroups. So in this video we're going to cover the epithelial tissue and look at their undergroup and how they're divided into the body. And we're also going to see the development of epithelial tissue. And at the end of this video I'm going to put some random epithelial tissue so you can try to pause the video and try to guess which cell types you're looking at. So talking about epithelial tissue, you're going to find two main types. So you have the covering epithelia and we have glandular epithelia. I'm not going to talk a lot about glandular epithelia as most of the times when we look at epithelial tissue you can think of covering epithelia. But I'm going to mention the most important structures you'll find in glandular epithelia that are important to know. So covering epithelia is called covering because it covers the structures you have in the body. The skin cells is going to be composed of the epithelial tissue. You're going to have the outside surfaces of organs going to be epithelial tissue, and also going to line the internal surfaces of organs. So as an example of that, if you look at this intestines right here, you're going to see that you can see epithelial tissue covering the external surfaces of the intestines, but it's also going to line the internal surfaces of it. You're going to have different types of epithelial tissue covering and lining, but they're all going to be epithelial tissue. Now when you talk about glandular epithelia, you're gonna think of glands, and when you think of glands, you're gonna think of either exocrine or endocrine, but you're gonna also see them in single cells like goblet cells. So an example of glands like this one, this is a sweat gland, consists of epithelial tissue around the glands right here. And so if we would look at it in a histological perspective, you would see that the internal structures of the intestines would look like this. You can see the villi right here, and you can see epithelia lining the surface of the internal part of the intestines. When we talk about the glandular epithelia, you can see that they're mostly going to look like this in tissue slides, where you're going to have the epithelial tissue lining up in a circular motion. Here, this is just a cross-section of the gland. Now, when we look at epithelial tissue, we need to know its development as well. So it all starts when a sperm fertilizes an egg and it becomes a zygote. And that the zygote is going to divide a lot and become a ball of cells called blastula. And blastula is going to keep dividing until it becomes something called gastrula. And then gastrula is going to keep dividing, and it's going to divide in different ways. You're going to have something called ectoderm. Ectoderms is in the name. Ecto means outer layer. So ectoderm, mesoderms, middle, and endoderms, internal. So the gastrula is going to divide into different body tissue types. And epithelial tissue is actually found within all these different derms. You're going to have it in ectoderm, mesoderm, and endotherm. You're going to find it in ectoderm, outer layer, you're going to find it in the skin. In mesoderm, the middle layer, you're going to find it in the tubules of the cell of the kidneys, for example. And in endoderm, you're going to find it covering the lung cells, the alveolar cells. We're going to look at it later on. So epithelial tissue may present different specialization along their various surfaces. So when we're talking about the apical pole, apical is a free surface. You're going to find it on the top of the epithelial tissue. And when we're talking about the lateral surface, on the lateral surface you're going to find some other type of specialization. Here you're going to find what is called the terminal bars, which you're going to find junctions. Junctions are going to be just two cells joined together. Junctions are really important to know because that's what gives the epithelial tissue one of its major functions. Because as you're studying epithelial tissue, you're going to see that epithelial tissue are really tight packed together. Now, and another thing we're going to look at is the basal part. And the basal part you're going to find at the bottom of the epithelial tissue, because you're going to find at the bottom every epithelial tissue, no matter what type of epithelial tissue you're going to study, you're going to always find what is called the basal membrane. And I'm going to talk more about the basal membrane in a bit. But first, let's look at the lateral surface first. On the lateral surface, you're going to find three types of junctions. The two cells are going to connect with each other in three different ways. So first, you have what is called tight junctions. And tight junctions, another name for it is going to be zonula occludens. And if you imagine you have a cell here, right? Here's one cell and here's another cell. If you can imagine one cell here, another cell here, and they're both going to have plasma membrane. And inside the plasma membrane, you're going to have transmembrane proteins. And that is what tight junctions is. It's going to be those transmembrane proteins that contact with the other cells' transmembrane proteins and form those tight junctions right here. And you're going to have a lot of different transmembrane proteins that form these tight junctions. But the most important transmembrane proteins you're going to find forming those tight junctions is going to be what is called the claudins. The claudins. Claudins. The claudins. And claudin is going to be the biggest participant when it comes to building this physical barrier. And these tight junctions are going to be so tight there's not going to be any room for water to pass through. They're just going to bounce off. And you're going to find most of the tight junctions in places like the bladder. You're going to find a lot of them in the bladder. You're going to find a lot of them also in the intestines. The intestines. It's really important to have these tight junctions when it comes to the bladder, for example, because you don't want the urine in the bladder to start leaking inside your body. And so it's called zonula occludens. Whenever you see the word zonula, that means that it's going to be bands that completely surrounds the cell. Now remember, the cell is three-dimensional, right? So these bands are going to surround the whole cell, making sure that there's no leakage of any type of liquid. The next type of cell junction you're going to find is the gap junctions. And these gap junctions are going to form these membrane channels right here. And through these membrane channels is going to pass water and ions. And you're going to find most of these type of gap junctions in tissues that spread action potential. The heart is going to have a lot of these membrane channels because the heart needs to keep pumping all the time, and it's able to communicate really well through these membrane channels. And these gap junctions, you're going to find them another name as well as communicating junctions. Now the third type of lateral junctions you're going to find in epithelial tissue is going to be adhesion junctions. These have two types actually. You're going to find them as zonula adherens or macula adherens. And here you're also going to have, if you imagine one cell here, right, and one cell here, and they're really close to each other. And what's going to happen here is that you're going to have those fibrin filaments right here that connects with this cytoplasmic plaque of transmembrane proteins. And then you're going to have what is called the cadarins that are going to project out and then fit with the cadarins of the other cell. And here's another picture just to show you visually how it works. You're going to have the filaments of the cytoskeleton right here, and here you're going to have the cytoplasmic plaque right here, and then the cadarin molecules that connect with the other cadarin of the other cell this way. And if you notice in the name, this one's called the zonula adherens. That means that this type of adherence is gonna completely surround the cell as well. Now the other type of adherence you're gonna find is macula adherent. Macula means that you're gonna find it on some spots in the cell. You're not gonna find it surrounding the whole cell, just on spots around it. And it does that through what is called the desmosomes. And to visualize this one, if you look at this picture right here, you're going to see that you're still going to have those filaments of the cytoskeleton. You're still going to have those plaques right here, right? Those plaques right here. And again, here you see that the macula adherens is going to connect in the same way as zonal adherens, but this time you're going to have other proteins involved. This time, this binding right here, these are called the desmosomes, right? These desmosomes. And the desmosomes in here are going to be made out of the desmoglein and desmocolin. And desmoglein is going to be also cadarin proteins. These are in the cadarin family of cadarin proteins. And to visualize this in a histological perspective, you're going to see really small projections out of these small cells. And these are going to be desmosomes surrounding the cells, attaching them together. The desmosomes are going to be a really strong type of cell junction that you're going to find them in places that are constantly going through stress, like the skin, for example. You're going to find a lot of desmosomes in the skin. And those are actually so strong bindings that if you were to pull the tissue really hard, these type of junctions is not going to tear. What is going to tear is the cell itself. So that's just going to give you a little picture of how strong these type of junctions are. And another thing I wanted to mention when it comes to desmosomes is that the distance between those two cells connected together is going to be a little further away than all the other type of junctions, and the distance between those two cells are going to be roughly around 30 nanometers. So it's going to be space for water and ions to go through freely, go through these type of cells like that. Alright, so that is the three type of junctions you're going to find when it comes to when you're looking at the lateral perspective of the epithelial tissue. Now, if you look at the basal pole, in the other hand, now remember I said that the epithelial tissue are so tightly packed together like this. Actually, it's so tightly packed together that there's not going to be any place for blood vessels or capillaries that are going to fit in between them. So they're going to need their nutrients in some other way. And that nutrient they get through what is called the basal membrane, right here, the basal membrane. And the basal membrane is going to work in two ways. It's going to work as anchoring membrane. So all those epithelial tissues are going to be anchored to this basal membrane. And no matter where you are in the body, no matter which type of epithelial tissue you're looking at, you're always going to find the basal membrane. You know, even though you're looking at glandular epithelia or lining epithelia, you're still going to find the basal membrane. And another thing you're always going to find is some type of connective tissue under the basal membrane. You're nearly always going to find those connective tissue under the basal membrane. And the connective tissue is going to have blood vessels inside it. So remember I said that the epithelial tissue are not going to have access to any blood vessels at all. So they need to get their nutrients in some way. How do they do that? They do that through diffusion of nutrients through the basal membrane. You're going to have nutrients going out that way. But you're also going to have waste products from the epithelial tissue go into the bloodstream so that then they can be secreted out from the body. So that's how they mainly stay alive. So the basal membrane is also going to work as a nutrient provider. All right? Nutrients. And if we were to zoom really close into the basal membrane, you're going to see that it consists of different laminas. It consists of lamina lucinda and lamina densa, which both may go together and make the basal lamina. And I've heard a lot of people confuse the basal lamina with the basal membrane, so don't do that mistake. The basal lamina is what the basal membrane is consist of. So the basal membrane is consist of the basal lamina and also the reticular lamina. So those two together is going to form the basal membrane. All right? And so the main components of the basal lamina is going to be the type 4 collagen. You're going to find a lot of that in the basal lamina. And so the basal lamina is going to be connected to the underlying connective tissue or underlying reticular lamina by the, it's going to be connected right about here, by the type 7 collagen. Type 7 collagen. And when I said underlying connective tissue, I meant that when you look inside the reticular lamina, you're going to find some connective tissue cells. That's going to make the reticular lamina cells, connective tissue cells. Alright, so when you're looking at the basal pole, you're also going to find another structure, and that's going to be what keeps the epithelial tissue connected to the basal lamina. They're going to do that through what is called the hemidesmosomes. So if you remember, I talked about the desmosomes. The hemidesmosomes are going to be, or we call them half a desmosome. And it's half a desmosome because it's not connected with another cell. It's connected with this type 4 collagen right here. And if you remember when I talked about the desmosomes, you're going to find the same structures here as well. You're going to find the filaments of cytoskeleton right here. And you're also going to find this plaque. So just like the desmosomes, hemidesmosomes are also going to have plaques. But these plaques are made by transmembrane proteins called integrins. And integrins is going to have receptors for the type 4 collagen in lamina lucinda. And just like the desmosomes, these bindings are really, really strong that if you were to pull the epithelial tissue away from the basal membrane, you're most probably not going to break the hemidesmosomes. What you're going to break is the cell itself. So those hemidesmosomes are really effective in keeping those cells together with the basal membrane. And so if we would look at all this in a histological view, we see basal membrane right here, right? Here's basal membrane. And here you're going to find those epithelial tissue. You're going to find different type of epithelial tissue here. I'm going to talk more about them later. But what I wanted to highlight is that under the basement membrane, you're always going to find those connective tissue right here. And inside the connective tissue, you're going to find blood vessels. Alright, so keep thinking about that when you look at epithelial tissue. You know, that's one way of orientating yourself when you look at slides of epithelial tissue. Always look for the basement membrane, and then under the basement membrane you're going to find connective tissue, and above it you're going to find different type of epithelial tissue. Alright, so that is the different characteristics you'll find on different sides of epithelial tissue. Now, as we go further and look at the classification of epithelial tissue, you're going to see that we divide them mainly into two types. You either find them as simple epithelium or stratified epithelium. So what does that mean? That means when you look at epithelial tissue, you're going to find that basal lamina again, right? That's basal lamina. When it comes to simple epithelium, that means that the type of epithelia you're looking at only going to have one layer. Right? Only gonna have one layer. When it comes to stratified is another word for many. So when you see that epithelial tissue has many type of layers, that's gonna be stratified. Stratified is gonna be two or more layers. So one thing that is important to know as well is that even though these cells are far away from the basement membrane, as you see here, they can still get oxygen and nutrients through diffusion. But there are some stratified epithelial tissue where cells do get too far away from the basal membrane and will eventually die due to lack of oxygen and nutrients. I will talk more about that in a little while, but keep that in mind during the video. Alright, so simple means one layer and stratified means two or more layers. But there's another way to classify epithelial tissue, and that is by looking at the shape of the cells as well. So there are three common types of epithelial tissue. We have squamous, cuboidal, and columnar epithelium tissue. So the first one is squamous, and squamous you'll find it as simple. And if you find it as simple, you will call this simple squamous epithelium tissue. So for instance, if you have the basal membrane here, right? And then you have these flat, thin, simple squamous epithelium tissue on top of that basal membrane. They're gonna look like flat pancakes on that basal membrane. But if we would pile up those simple squamous epithelium tissue on top of each other, we will get stratified. We'll call this stratified. Alright, we'll get stratified squamous epithelium tissue. Alright, so then we have cuboidal. And cuboidal, these cells, you'll find them cube-shaped. Alright, cuboidal typically come as simple. You will rarely find them as stratified in the human body, but I'm going to show you an example of when we actually do that, when we actually find cuboidal cells as stratified. So then we have columnar, and columnar you're going to mostly find these as simple as well. Since these cells are really tall, they don't really need a lot of layers on these for them to serve their function. Alright, so when we're talking about cuboidal, these cells you'll mostly find in areas with secretion, like glands. And you'll also find these in the renal tubules in the kidney as well. And when it comes to cuboidal, you will find these in absorptive areas because these cells are really tall. They can catch a lot of nutrients they want to absorb. Alright, and then we have squamous epithelium tissue. These cells you'll actually find everywhere in the body. You'll find them as covering epithelial tissue, and you'll find them in absorptive areas as well. Alright, so another thing you will notice when you look at all these three cells is the nucleus. You'll see that the nucleus is going to be flat and elongated, while when we go over to the cuboidal are going to have a rounded nucleus. And when it comes to columnar are going to have elongated nucleus, and you're going to mostly find them at the lower 1/3 of the cell. Alright, so that's essentially how you classify epithelium tissue. You look at how many layers of cells we have, and you look at the shape of the cell, and you also look at the shape of the nucleus as well. Alright, so that is the three types of epithelial tissue you have in the body. Now if we start with simple squamous epithelium tissue. Before I start, I just want to mention that all these slides I used in this presentation I got from this website right here. This website belonged to the Department of Histology in the University of Michigan. I found this website really useful when it comes to studying histology, because in this website you can actually search for whatever organ you want to see, and it gives you a lot of different slides from different organs. And the good thing here is that you can actually search for what type of cell you want to look at, and it gives you a lot of different organs and slides that actually shows that specific cell you're looking for. So I'll put the link in the description if you want to use that. Alright, so now starting with simple squamous epithelium. So these cells, as mentioned, these are going to be flat cells, right? And since these cells are flat, you're going to immediately think that protection is not their main function. These type of cells, you're going to mostly find them in areas where the secretion and absorption, and also where filtration occurs. So if you use this picture as an example, I don't know if you're familiar with the alveoli. This is going to be alveoli right here. Alveoli, you're going to find them in the lungs. So if you say, here are the lungs, right? You breathe in air, and that air is going to go into your lungs, and then into what's called bronchial tubes, and then into those air sacs called alveoli. And this is where the exchange between oxygen and carbon dioxide is going to happen. You'll find a lot of capillaries surrounding these alveoli. Alright, so if you look at this picture again, you will see that the simple squamous epithelium is going to surround these air sacs right here, these alveoli. Then remember, under the simple squamous epithelial tissue, you're going to have what is called the basal membrane, right? Alright, since you're also always going to find epithelial tissue resting on basal membrane. Alright, so when you breathe in air, those alveoli is going to be filled with air, right? All those air sacs are going to be filled with air. What's going to happen is that the good thing in having those thin, small, simple squamous epithelial tissue is that they're going to allow rapid exchange of oxygen and carbon dioxide. In that way, this exchange right here will happen really, really fast. And then if you would look at these cells from an apical view, I'll tell you a little more about what apical means. But apical mean is, so if we have the basal membrane right here, and then we have epithelial tissue, apical part is the surface of it. So if you look from the top of the cell, the apical view will look like this, right? You can see that these cells have different shapes right here. Alright, and they're usually really easy to spot because they don't have any cuboidal shape or columnar shape like the other two types of epithelial tissue we have. Alright, another thing you gotta notice here is that these type of cells, the simple squamous epithelium, is gonna cover the visceral organs. And then you can immediately think, these cells are really thin, so what's the use in having them cover the visceral organs if they're not able to protect the organs? Alright, so let's say you have the heart right here, right? And then here you have intestines. And then you have that simple squamous epithelium tissue covering those organs right here, right? And that was the good thing with that simple squamous epithelium tissue is that they're going to form what is called serous membrane. Alright, and the good thing about having that serous membrane is that it's going to make sure that the surrounding area stays wet. Alright, so think about this a little bit. What does the heart and the intestines have in common? They're both going to be in constant movement, right? So the heart needs to keep beating to keep that heart circulation going, and the intestines need to keep moving to digest that food you eat. And that's a good thing in having that wet surface, because it reduces the outside friction when these organs move. Alright, so that's why we have simple squamous epithelium tissue covering those visceral organs. Now, another place you're going to find simple squamous epithelium is at the glomerulus of the nephron in the kidney. I don't know if you're familiar with the nephron in the kidney, but remember the main function of the kidney is filtration of the blood and producing urine, right? And they're able to do that by those nephron right here. So imagine this is the head of the nephron, and then you have the body of that nephron, the tubules right here. Right, that's going to lead the newly filtrated urine down to your urinary bladder. Alright, so one thing to remember here is that, remember you have 55% of your blood is water, right? And then you have 45% of the blood is going to be cells, but only a certain 5% is going to be sugar, proteins, and also you're going to have waste products containing nitrogen, right? And eventually you want to get rid of those nitrogen. What's going to happen is that you have these blood vessels right here, right? They're going to come into that head of the nephron, the glomerulus, and they're going to turn into capillaries, and those capillaries are going to be thinner, right? And because they're going to become thinner, that blood is going to come into your capillaries with a high velocity, right? The pressure is going to increase. And that's a good thing in having these simple squamous epithelium tissue lining that glomerulus right here, is because everything that is small enough to go through those cells are going to be filtrated into your urine. Alright, so these simple squamous epithelium tissue allow for rapid absorption of those substances right here. And in this picture right here, you're gonna see the glomerulus right here. And you're gonna also see those thin epithelium tissue right here, those thin simple squamous epithelia. And remember, epithelial tissue is always gonna rest on the basal membrane. Alright, so here we see the simple squamous tissue right here. And you're also going to find capillaries in the middle here. So what's going to happen is that substances are going to be filtrated through those simple squamous epithelial tissue. Alright, so that was simple squamous epithelium tissue. The next one is the simple cuboidal epithelium tissue. Remember, these type of cells you'll find in areas where secretion, absorption, and filtration occur. So an example of that is the renal tubules. Remember we said that the head of the nephron is going to be simple squamous epithelium tissue, the glomerulus. And so if you look at the glomerulus right here, everything after the glomerulus is going to be a simple cuboidal epithelium. And everything in the cuboidal is going to be simple squamous epithelium. Alright, and these structures right here, you can see that they're cube-shaped. These structures are going to be a cross-section of the renal tubules of the nephron. Alright, so we're still in the kidney, remember that. Alright, and another thing, one way to orientate yourself if you're looking at a renal tubules is, you have a proximal tube, you have the loop of Henle, you have the distal tube. Alright, so we classify the tubes into different types. And as you see here in this picture right here, you will see that this one is taken from the proximal convolute tube, this one right here. And how do you see that is that the simple cuboidal epithelium in the proximal tube are the only cells in the renal tubules that's going to have these small hair-like right here. You can see very small projections coming out from the simple cuboidal epithelium. Alright, so let me just remove everything first. Alright, so here you see the basement membrane again, right? Remember, we also was going to see basement membrane. And here you see the cuboidal cells. You can see that they have a rounded nucleus and you can see that they're cube-shaped, right? And the good thing in having those cuboidal epithelium tissue in your renal tubules is that, when blood is filtrated and it comes that way, that direction, as I said, everything that's small enough to be filtrated are going to be filtrated. But we don't want to get rid of all the sugar. We don't want to get rid of all the proteins. We just want to get rid of the waste products, right? So what happens is that this pre-urine, we call it pre-urine because it hasn't been refiltrated yet. Some of the sugar and the protein and the ions and water even are going to be reabsorbed into the bloodstream again. And that's a good thing in having those microvilli inside our proximal convolute tube, is because in the proximal tube you will receive newly filtrated pre-urine and they're most probably going to contain a lot of proteins and water and stuff. So you need something to assist the reabsorption, and that's those microvilli is going to do. That's why you find the microvilli together with the simple cuboidal epithelium tissue when you look at the proximal tube of the nephron. Alright, so remember these are called microvilli. These are called microvilli. And microvilli are typically going to be about 1 micrometer. Alright, so another place you're going to find simple cuboidal epithelium is usually in the ovaries. You can also find them in part of the eye, but you're also going to find them in glands. Alright, so you're going to find them in endocrine, exocrine gland. I'll talk more about that later. Alright, so that is simple cuboidal. Now going over to simple columnar epithelium. These cells are going to be highly specialized cells in absorption. Remember I said that? That's because they're very long cells. Alright, so if you look at this picture, for example, if you think of absorption, that's the main function of the simple columnar epithelium, you will think of the intestines because you need to absorb the nutrients you eat, right? So this picture right here, you'll see a cross-section of the intestines, and here you'll find the lumen. The lumen is mainly going to be nothing right here. That's just going to be a hollow opening or space. That's going to be a space for the food you ate. Alright, so if one thing you also notice right here is those finger-like projections you see right here. You'll find these mostly inside the um, inside the intestines. So if you go ahead and zoom in on one of them, it would look like this, right? We'll see that it looks like a finger. And these finger-like projections are called villi. And remember, as I said before, you have the basal membrane right here. And under the basement membrane, you have loose connective tissue, and you have blood vessels as well, so that the nutrients you eat is gonna end up in the bloodstream. Alright, and here you see the tall columnar cells. You can see that they're tall, and you can see that the nuclei are located in 1/3 lower part of the cell, right? A small part right here. And again, these are highly specialized cells because they're tall, and that's a really good thing in having these tall columnar epithelium inside your intestines, because they're tall and they can catch a lot of different nutrients right here coming through. Another thing that's important to mention when coming to the villi is that, on the surface of the villi, you're going to also see small hair-like projections, the microvilli. You will find a lot of microvilli inside your intestines lining these villi right here, and they're also going to assist with absorption. You have the basal membrane here, right? And you have these tall columnar cells, and you have these microvilli peeking out from the apical surface of the simple columnar epithelium. Alright, remember, these are going to be mostly 1 micrometer long, and they assist with absorption. And how do they do that? If you imagine this is the microvilli, right? And when this microvilli comes in contact with nutrients, what's going to happen is that the fibers inside the microvilli is going to contract, and it's going to lead the nutrients towards the simple columnar epithelium. Alright, so it's a really good thing in having those microvilli inside our intestines. Alright, so another place you'll find them is in the uterine tubes. Alright, so that is the simple columnar. The next one is pseudo-stratified columnar epithelium tissue. That was hard to say. But if you break the words down, pseudo means false, right? False. Now remember, when you see the word stratified, that means multi-layer, right? We call this pseudo-stratified because when you look at it in the microscopes, like you see right here, this tissue looks like multi-layer, but they're really not. So basically, every cell right here is going to originate from the same basement membrane. But not all the cells are going to have the same height. There are some cells that are going to be shorter than the others. And that's why it looks like multi-layer, because these nuclei, you'll find them in different levels in the epithelial tissue. Alright, and you'll mostly find these pseudo-stratified columnar epithelium in conducting airways. Alright, esophagus would be an example of that. Alright, and the good thing in having those pseudo-stratified columnar epithelium lining the conducting airways is that you see those small hair-like projections you see right here? These hair-like projections are going to be called cilia. Cilia are going to be longer. Remember, I said that microvilli is mainly going to be 1 micrometer long, but these cilia, these are going to be longer. These are going to be 10 micrometer, about 10 micrometer. And if you look at the structures inside the cilia, you'll mostly think of this equation right here, 9 times 2 plus 2. And the reason why is because you'll find 9 pairs of microtubules located in the peripheral part of the cilia. Peripheral means that they're just going to be located at the surrounding end of the cilia. Alright, so here you see 9 pairs. So these are 1, 2 microtubules, right? And these 9 pair of microtubules are going to be mainly found surrounding these 2 centrally located microtubules. That's why plus 2 is because you have 2 microtubules in the centrum. So again, the structures of cilia is 9 pair of microtubules surrounding 2 centrally located microtubules. Alright, so another structure you're mostly going to find in pseudo-stratified columnar epithelium is those goblet cells right here. You're going to find something called goblet cells, and goblet cells are going to be simple cell glandular epithelia. I'm going to talk about it more later, but that's also one thing I forgot to mention when it comes to the simple columnar epithelium in the intestines. These are also going to have these goblet cells. And the good thing about these goblet cells is that they're going to produce what is called mucin, right? And that mucin is going to react with water, and it's going to become mucus. Alright, so you're going to have a layer of mucus right here as well, together with the cilia. And the good thing in having those mucus and the cilia in your conducting airways is that when you breathe in air, right, what it does is that it works as filtration as well. and it's also gonna warm up the air you breathe in. Alright, so in that way it's less chance to get sick. And another thing is that when you breathe in something, these cilia is gonna catch that, right? And what happens when those cilia catches something, we're gonna start coughing a lot. And when we cough, those cilia is gonna assist us in removing that thing we just breathed in. We're gonna cough, and it's gonna come out as mucus. You can either spit it out or swallow it. Alright, so that is the pseudo-stratified columnar epithelium. Now, if we go over to something called stratified squamous epithelium, now remember I talked about the single squamous epithelium where it only have one layer of cells on a basement membrane. This one is stratified. This one's going to be more layers of squamous epithelium on the basement membrane. So stratified squamous are divided into two parts. You either have keratinized or non-keratinized. A keratinized epithelium you will mostly find in dry areas, while non-keratinized you'll mostly find in wet areas. Alright, so what's interesting about this stratified squamous epithelium is that the deeper cells you'll find closest to the basement membrane are going to have a cuboidal or columnar shape. But as they divide a lot, it's going to move old cells upwards, and then you're going to see that the outer layer are going to be stratified epithelium, right? Another thing when it comes to a keratinized stratified squamous epithelium is that these cells gets reinforced when they're down here at the basal layer. And they're going to be reinforced with something called keratin. That's why they're called keratinized. Keratin is going to be a protein that provides a strength to the epithelia. It's going to protect it from damage or stress. Alright, so another function keratin has is that it's extremely insoluble in water and organic solvents. And you'll mostly find these keratinized stratified squamous epithelia in the skin. Alright, so if you can imagine, this is the outer layer of the skin, right? And this right here is going to be a lot of dead stratified squamous epithelia filled with protein keratin, right? And keratin, as I said, is going to be extremely insoluble in water and organic solvent. And that's the main reason why our skin is waterproof, right? It's because of those keratin right here. Alright, so now if you start with non-keratinized squamous epithelia, so again, think of these as wet areas, alright? Think of these as, when you see a stratified squamous non-keratinized, think of these as lining the mouth or the esophagus or the small intestine, and also the vagina is going to be lined by these stratified squamous non-keratinized epithelia. Alright, so again, we got the basal membrane right here, right? And we got loose connective tissue filled with blood vessels. Alright, so if you look at the cells closest to the basal membrane right here, they're going to look the healthiest. And you know, knowing the function of the basement membrane, it's going to be logical because these cells are going to be closest to the source of oxygen and nutrients, right? Right? And another function epithelial tissue has, I forgot to mention it earlier, but they're what we call high turnover type tissue. That means that they're going to have a rapid rate of mitosis. So these cells are going to divide a lot, and old cells are going to be pushed out towards the surface. Alright, so they divide a lot and old cells are pushed out. And cells at the apical surface here are mostly going to be replaced every 2 to 3 days, right? This picture is going to be the lining of your stomach. So the surface of your stomach are going to be replaced every 2 to 3 days due to this rapid mitosis happening in the epithelial tissue right here. Alright, so one thing you notice is that the further away these squamous epithelia gets from the basement membrane, the shape of the cells are going to change, right? You can see that the cells up here looks different from the cells down here. These cells up here are slowly dying. That's why the shape is different. It's because they're far away from the source of oxygen and nutrients. But you know, on the apical surface here, these cells are still going to be alive. And that's the biggest difference between keratinized and non-keratinized epithelium. In keratinized, these cells at the apical surface are going to be dead. But in non-keratinized, these cells are still alive. They're on the last stages of their lives, but they're still alive. Alright, so when it comes to keratinized stratified squamous epithelia, remember here are the basal membrane, right? And here you see loose connective tissue and blood vessels around here, right? So these cells are keratinized, meaning that they have the keratin protein. Right? So these cells are going to be keratinized. These cells are going to divide a lot, and old cells are going to be pushed up to the surface like that. And when it comes to the surface, these stratified squamous keratinized epithelium are going to be dead. And they're going to pile up with a lot of keratin in them. And again, that's mostly the reason why our skin is water resistant, right? And again, when you think of keratinized stratified squamous epithelia, think of dry areas like your skin. Alright, so that is stratified squamous keratinized epithelium. Now the next one is stratified cuboidal epithelium. And when it comes to stratified cuboidal epithelium, remember I said that these are going to be rare to find. You're mostly going to see a stratified cuboidal epithelium as 2 to 3 layers maximum, as you see here. And these are going to be located in sweat glands and in intestines of the male. Alright, so that is stratified cuboidal epithelium. Here you see a picture of a sweat gland. Alright, so that is the stratified cuboidal epithelia. Now going over to transitional epithelia. At transitional epithelia, these are going to be cool cells because these cells are going to change their form according to the stress they receive. So one place you'll find these cells is in the urinary bladder and also in the ureter. So if you don't know what the ureter is, here you see the kidney, right? And here you see the bladder, and the ureter is going to be what leads the urine from your kidney to the urinary bladder. Alright, so that is ureter. And they change their form according to how much urine they are leading to the bladder. And here you see a cross section of the ureter. Here you see that this one is stretched and this one is relaxed. And transitional epithelia is going to be located at the inner surface of the ureter. Alright, so if you go ahead and zoom in on these cells, you will see that these cells are going to look like this. Alright, so transitional epithelia is not going to be cuboidal, it's not going to be columnar, it's going to be just transitional epithelia because these cells are going to change their form, right? And one thing you're going to notice here is that these cells right here in relaxed form are going to look like a pear. It's going to have a pear-shaped cells right here, alright? And their nucleus are going to be rounded and really big. But as soon as a lot of urines it comes down this direction, these cells are going to stretch, and when they get stressed, these cells gets flattened, and also the nucleus, it goes from round to flat. Alright, and that's a good thing in having these transitional epithelia lining those places that are receiving a lot of stress, because these can change their form. Alright, so that is the transitional epithelia. Alright, and then we have glandular epithelia. Glandular epithelia is going to be formed by cells specialized to produce secretion. And we're going to mostly find glandular epithelium as two types. You're going to find them as multicellular, when exocrine and endocrine is going to be an example of that. And you'll also find them as unicellular, as goblet cells. And remember, I talked about goblet cells when I talked about Pseudo-Stratified epithelium. And you're also going to find them in the intestines between the simple columnar epithelium. And so this is the goblet cells. That's how it looks like. This is the structures inside it. Right, so here you see the nucleus with their DNA right here. Alright, and remember the main function of cells is to produce proteins, right? You got these mitochondria here that fuel the process of protein synthesis. Alright, so you're going to have a lot of rough endoplasmatic reticulum as well. And you're going to have a lot of Golgi apparatus as well, which packs the proteins and sends this away. Alright, and now we got all these packed proteins ready to go out from the cell. And that packed proteins, remember as I said that the cell produces mucin, and comes in contact with water and becomes mucus. Right, so mucin comes in contact with water and then becomes mucus. Alright, and you'll find these goblet cells between epithelial cells, as I mentioned. Now I'll just give you a little challenge here. Can you guess what kind of epithelial cells you see right here? I'll give you 3 seconds to think about that. 1, 2, 3. That's going to be pseudo-stratified epithelium, because you see that all the cells are still going to be in contact with the basal membrane, but the nucleus are going to be found at different levels. Alright, so that is unicellular. Now going over to exocrine and endocrine, the multicellular glandular epithelium. Alright, so we classify glands based on their secretion, right? It's either going to have endocrine or exocrine glands. And endo means within. So the products of endocrine is going to go within the body, in our bloodstream. And the products of exocrine is going to go outside, right, to any surface where these glands are located. Alright, so imagine endocrine are glands that produce hormones, right? You're going to have this cell right here, but these cells are going to be ductless. They're not going to have any ducts. So what happens is that when these cells are going to produce hormones, right? And then these hormones are going to diffuse out to the body tissue fluid, where you're going to have blood vessels that eventually are going to catch those hormones and then send them away over a long distance. Alright, so that's another thing to think of when you think of endocrine, is that their secretion is going to travel a really long distance to another part of the body. But when it comes to exocrine, on the hand, exocrine is going to have ducts, right? These ducts right here. It's going to have a duct and it's also going to have a secretion part. And I just want to mention that, remember I said that cuboidal cells are mostly going to found in glandular epithelia as well. Here you see those cube-shaped epithelia right here, all right? They're going to lie in both exocrine and endocrine glands. Alright, so these cells are going to synthesize their secretion. Alright, so this is interesting. You'll find another type of epithelia surrounding these glands. And these type of epithelia is called myoepithelia. Alright, so it's called myoepithelia. And it's called myoepithelia. When you see the word myo, you will think of muscles. Right, so these epithelial cells, there are specialized cells that can contract, and when they contract, they're going to squeeze this duct and lead their secretion up to the surface. Alright, so that is mainly the two type of multicellular glandular epithelia we have in the body. And a very known example of these two glands is the pancreas, which is a mixed gland. And a mixed gland means that it contains both endocrine and exocrine glandular epithelia. So I don't know if you're familiar with the functions of the pancreas, but about 98% of the function of the pancreas is going to be exocrine, right? Exocrine. And that's because you're going to have a lot of exocrine glands leading to the intestines, assisting the digestion of the food you eat. Or so that's 98% of the function that the pancreas has. Now the rest of the 2% of the function in the pancreas is going to be endocrine. Right? And when you think of endocrine in the pancreas, that's going to produce what we call insulin and glucagon. Alright, and these two hormones are going to be found in the beta cells in the pancreas. So, alright, so that is the different glands we have in the body. And when you think of exocrine, you're going to see, oops, let me just remove that. You have the gastric gland where you're going to find those straight tubules. You're going to see here, you're going to find the secretion part here and the duct part here. And you can also find these tubules as branched. You're going to find the secretion part branched right here and a duct. And it's really important to have these branched when you need rapid production of the secretion they produce. And in the pancreas, you're going to find the acinar glands. These glands are going to have, or the secretion part of those glands are going to be bigger and fatter. And if you think of the pancreas, you'll think of 98, as I said, 98% of the pancreas is to assist the digestion of the food you eat. So you need to produce a lot of this secretion right here. And you're also going to find them in salivary glands. And if you think the salivary glands, you need to produce a lot of saliva. That's why it's a good thing to have a branched type of acinar secretion part. Right? So that's mostly how we classify those glands right here. And another way to classify it is that you see that duct right here. If that duct would be shorter, we would call this alveolar duct. Alveolar duct. It will look like an alveoli in the lung. And if this gland has a long duct, it will be tubular. Alright, another thing I wanted to mention is that when you look at a histological slide of endocrine, you will see that these cells are pretty light compared to the surroundings. So these cells are quite easy to spot if you ever see endocrine glands. Alright, so that is the glandular epithelium. Now one more thing I wanted to talk about really quick is the neuroepithelium. A neuroepithelium is in the name neuro, that means they are with the nerve cells. And here they serve as supporting cells for the nerves passing through. You're going to mostly find them in the roof of the nose, right? Here you see the lamina cribrosa of the ethmoid bone and the olfactory bulb with their nerve cell that sense smell. You can see those neuroepithelium in between those nerve cells. And you know, even though you look at neuroepithelium, you're still going to find that basal membrane right here with the basal cells. Now another place you'll find the neuroepithelium is in the tongue. You'll find them protecting the taste receptors right here. You know, you're still going to have basal membrane and you're going to have these neuroepithelium protecting these taste receptor cells right here. Alright, so that is all the epithelium I wanted to talk about. Now here you see 3 histological slides of epithelial tissue. Now can you pause the video and guess which type of epithelial cells you're looking at? Alright, so I will reveal the answer in 3, 2, 1, there you go. So if you got these correct, you actually learned something. Alright, so that was the video about the epithelial tissue and I hope this was helpful.